Hello, my name is Swasti. A genetically modified microbe, or GMM, serves many purposes, such as cleaning up an oil spill, detecting heavy metals in water, eliminating harmful diseases, and more. These GMMs are beneficial to society, but some can cause unforeseen problems if not properly controlled. Biocontainment strategies have come a long way since the discovery of engineering DNA, but they are not perfect. Mutations within the biocontainment system can occur, rendering the synthetically installed safety mechanism useless. The goal of every researcher designing a biocontainment mechanism is to minimize the mutation or escape rate. Though this rate can be notably decreased, there will always remain a small number of escapees. There are two commonly used biocontainment strategies available: Oxatrophy and kill switches. Oxatrophy involves taking out an essential gene so the target organism is reliant on an external nutrient. This is helpful when the nutrient can be applied to a certain area to keep the organism alive, like in a bioremediation site. However, this strategy is not effective in cases such as cleaning up microplastics in the ocean. The organisms must survive outside of a limited area to effectively clean up the widespread microplastics. Kill switches are genetic circuits that respond to a signal molecule by translating specific regions of DNA, which results in the death of the cell, often by a toxin. These switches can be induced by various factors which further define the type of kill switch. Chemical-induced kill switches produce a deadly cell toxin in the presence of a specific chemical molecule. Optogenetic switches are kill switches induced by light. These switches are useful in containing a bacterium inside an environment that doesn't receive much sunlight, such as a beehive or underground. Temperature-induced kill switches ensure an organism only survives at certain temperatures, for example, in a warm mammalian gut or within freezing ice. Biocontainment systems have room to improve, and researchers are constantly searching for better ways to reduce mutation rates and horizontal gene transfer. Gene drives are the newest type of biocontainment mechanism, which allow researchers to control a target population. So far, this technique has only been efficient in a limited number of species, but is a beacon of hope in eliminating malaria-carrying mosquitoes. Gene drives spread alternative genes through a population to eliminate a certain characteristic. For example, the ability of mosquitoes to transmit malaria to humans. However, this type of biocontainment mechanism raises ethical issues regarding the potential of wiping out an entire species. This information is useful when choosing a safety mechanism for your engineered organism to prevent it from doing anything other than its intended purpose. SynBioHub and the iGEM registry are good registries to find specific parts on. Additionally, Ohio State's 2020 iGEM team has worked on biocontainment mechanisms, and more information can be found on their wiki page.